Well, thank you. First of all, thanks to the organizers and particularly to Vanessa for the invitation. Also to congratulate them for this nice meeting. It's spectacular, the audience that we have today and yesterday here. And the talks have been really nice. Yesterday very inspiring, today very educative. So thank you. And also I want to encourage you to continue supporting all the basic research, all the scientists that are around working on basic research on this disease, because this is very important for companies such as ours trying to develop therapies. We need basic research out there to allow us to attempt to discover a drug. Without basic research, there's no therapies, for sure. So being said that, I want to add something, is a disclaimer for patients particularly and for families. We are working on therapies, but we are still far, far away. When Vanessa invited us, we were a bit dubious about talking today here because sometimes this can create too much expectation. And I want to clarify that we are very far from a therapy. So being said that, I will talk about a potential way to treat CDG. We will talk about a kind of drugs that are named pharmacological chaperons. So I will give you a little bit of review of this, followed by a more divulgative way of explaining the challenges that we are facing. So let me just introduce briefly Minoryx. Minoryx is a Catalan company here from Barcelona. We are focused on rare neurometabolic diseases with a high medical need, and we are using different approaches there. This is the team from Minoryx. These are the founders, Joan, Xavier, and myself. And Pilar and Anna take care of biology and chemistry. And in the lab, we have Laura, Marc, and Aida. Also, a long list of collaborators. This is our highlighted collaborators. We have others. Sant Joan de Déu, University of Barcelona, University Autónoma of Madrid, IDIBEL, a company called Sagetis. And we are financed essentially through private investors and also public loans and some grants. So what is a pharmacological chaperone? Let's start there. So this is something that you are very familiar with. This is a disease. Imagine a disease where this is a monogenic disease. So that means that there's one gene that has a defect. The consequence is that there's one protein that is not working well. So there's a cellular dysfunction, which causes the disease. It's simple, or relatively simple. So in the case of CDG, of this CDG, the problem is the deficiency of mannose 1-phosphate. This substrate is not formed because this protein is not working well. So what can we do? We can either work downstream, working with palliative treatments or symptomatic treatments. We can administer mannose 1-phosphate. That could be a theoretical approach. The problem here, as Eva explained in other CDGs, this could be an approach. The problem is where this substrate has to go. Has to go to the brain, for instance. This is a complication. So it's not always possible, this approach. And the third way is let's try to recover the enzyme. If the enzyme is not working well, let's try to do it work well again. Gene therapy could be one strategy. Let's correct the gene. Let's correct the protein. This is enzyme replacement therapy. Or let's try to restore protein functionality. This is pharmacological chaperons. And this is what I will try to explain to you today. This kind of P, it represents a protein. In blue is a protein that works normally, has the normal structure, so it works as usual. This is an unstable protein with a mutation that makes the protein less stable than normal. Luckily for us, in the cell, we have a quality control that prevents of having errors on the production of proteins. But when we have a protein that has a mutation permanently, this quality control system degrades our protein permanently. So we have protein deficiency, we have a disease. If we add a pharmacological chaperone, is a kind of glue. You can imagine it as a glue. Is a small molecule that stabilizes this protein. So it prevents the machinery, the quality control machinery, to degrade the protein. So we keep functionality. So we have a potential way for recovery. This is nothing new, or it's quite new, but there are already other compounds in development or either in the market with this strategy, so it has a certain level of validation. And I will only add that in Minoryx we have a platform that allows us to identify this kind of compounds, but with a significant difference compared with the pharmacological chaperons that have been described up to date. The significant difference is that we identify compounds which are non-competitive with the natural substrate. And this has many implications in terms of safety and therapeutic window. I will not go into detail on that. This is the divulgative part, particularly for patients, that we have to discover a drug, but where do we start? This is the whole universe. But maybe you don't know that, but if we try to prepare just 10 milligrams of all the possible drugs, we require more than the whole mass of the universe. So we cannot explore that, obviously. The good news is that we only explore 0.0000, many zeros, 1% of the universe. So there's a lot to invent. But it's difficult for us to know where to start. And this is the first challenge that we have, where to start. The second challenge that we have is the protein. This is the three-dimensional structure of the protein. At the end of the day, it's just a shape with functional groups, but at the end of the day, it's a shape. We can compare this with the lock and the key. Imagine that the protein is a lock, the key is the drug. If you have a lock that you cannot see, you don't know which key you have to try. It's very difficult. But if you see the lock, at least you can say, well, is this planar key at least? And you can try among the planar keys. Drug discovery is the same. If we have the idea which is the protein and the shape of the protein, then this can direct us pretty much on the chemical space to start to explore. But then we go to the cell. The cell is much more complicated than just a protein. This is maybe too difficult to explain it today in a few minutes. So let's look at it like this. The cell is a factory at the end of the day. So it has many different employees doing many different things, some of them not really fundamental. So some of them quite important. One, this is not working well, nothing happens. But if this is not working, we have a problem. So this is the cell. But in reality, this people looks pretty different. But the real cell is more like this. So we have many different proteins, highly specialized, and they look very similar, some of them. And this is the selectivity problem that we have when we do drugs. So to get a drug that is selective, only targeting the protein that you want is not as easy as it seems. We have a compound, a drug that targets the protein. Great. We go to the brain, where, for instance, for CDG1A is important. We have the blood-brain barrier. This is a quite complicated thing again, so difficult to explain, but we can understand it like this. In the brain is where we control everything, so it's extremely well protected. There are all kinds of barriers, so we cannot go there, simply. The Pentagon is the same. Try to enter there walking, and you will not enter. So also, we think in consumption of resources probably is also similar, so they waste a lot of money also. But cells are not animals. So again, we reach the brain, but animals is another history. And animals are not humans, simply. So what do we know? We know many things. Fortunately, our physicians are in the room. They know many, many things, much more than this. So, but there are many other things that we don't know yet. So when we go with a drug that has been tested in cells, in animals, with great results, we cannot predict if this drug will be efficacious or toxic in humans. So we have to test it. We can have an educated guess, but we cannot know for sure. And this is drug discovery, essentially. So we start with the whole universe, and we go farther and farther up to the market. And because there are so many uncertainties, this is the reason why probably the pharmaceutical industry is one of the industries with the highest attrition rate. So only 1 of every 5 compounds that enter in clinical development really reach the market. And we need to test many, many compounds to have one of these compounds to be tested in clinical trials. So for patients, we are just in the beginning. How we do it in the chaperons, because this is general drug discovery, but how do we identify chaperons? The first thing is to look in the chemical space. There we use computational methods. This allows us to explore very efficiently a big part of the chemical space, not all, unfortunately, but a big part. Then we go to the lab, and we validate these computationally identified compounds using thermal stability in the case of pharmacological chaperons, enzyme enhancement, etc. Then we go to the cells, and these are these beautiful pictures with colors that we have seen today, also with green fluorescent protein, etc. To look for substrate levels, protein, also to start to assess toxicity. And then we have to go to the animals. And here we look to recovery of the enzymatic activity in the relevant tissues, and also to recovery of the phenotype, of the normal phenotype. And then to the clinical trials, where patients are in the center of the trial, but I have to say that in the particular field of rare diseases, patients, you are everywhere. As has been said this morning, without your help, the basic science that is required to do all these steps is not there. So where we are. We have a first project in GM1 gangliosidosis, a lysosomal storage disease, quite severe also. And in the CDG, we are close to the lead optimization stage. So as I said early, we have a long, long way. What we are doing. This is a project that we are doing in collaboration with University Autónoma de Madrid, Belén Pérez, and some of her colleagues that are in the room today. Belén and her group already published in the past that CDG PM2 is essentially conformational disease because there are many mutations that are conformational mutations. That means that they destabilize the protein. The protein is less stable than normal, so the protein is degraded, and because it's degraded, there's a deficiency that causes the disease. And at least one of the alleles in many patients is one of the folding mutations. So this means that theoretically, chaperons could be an alternative for this disease. This theory they demonstrated that testing more than around 10,000 compounds in collaboration with Bergen University by different techniques. With this, they identified several hits, and where we are now is working together to optimize these hits up to a candidate. This requires many things. It's a matter of optimization of potency, selectivity, toxicity, et cetera, et cetera. All the challenges that I said that we have to achieve before getting a real drug. So just to summarize, there are several mutations for which chaperons could be an alternative for this disease, although further studies are required. The primary studies suggest that we are going in the appropriate direction, but we have a long, long, long and uncertain way to go. We have many, many turns, ups and downs. We don't know if we will reach the end of the road. We cannot promise that. We don't know. What we can only say is that we are walking, so we will try. Let me finish also mentioning Belén, Celia, Patricia, and Alejandra from Autónoma de Madrid, that are the partners in this project, and all of you for your kind attention.